this is the last and final video of Unit 5. And in this video, we're going to look at this concept of osmosis. And I have a cool picture up here in the upper right-hand corner of my screen. And that picture is showing water. So osmosis has an integral part of water. So water moves in and out of cells just like other things move in and out of cells. But water, we give it its own name. We call it osmosis. And there's a couple of things that we need to remember when it comes to water. So that's what we're going to cover in this. Is osmosis is the movement of water inside and out of cells. So if we look here, in the middle, here's our semi-permeable membrane. And we notice in this semi-permeable membrane that there are holes that allow things to move through. Well, if we take a look at this salt here, this salt molecule is far too big to go fit through this little hole. So we notice that there is a lot of salt on this side. So we have, this would be our high concentration of salt. So here's our high concentration of salt. And over here is our low concentration of salt. Right, so the salt wants to move from high concentration to low. So the salt really wants to go across that cell membrane. But we notice that the salt cannot go through those things. So because of that concentration gradient, because there's a high concentration of salt over here and a low concentration of salt over here, and the salt cannot move, that's actually going to force water to move. And water is going to move in the opposite direction that the salt would want to go. So the water is going to move towards where there is more salt. And the water is going to want to dilute out that salt. This movement of water to an area of high concentration of water to low, so right over here, if we notice we have lots more water molecules, this would be a high concentration of water. High concentration of water. Yeah, you get what I'm saying. And over here is a low concentration of water. So the water wants to move from high concentration to low, but it wants to go where there's a high concentration of salt. Kind of an interesting idea. It wants to move in the opposite direction. So if we look over here, osmosis, it's another type of passive transport. Cells don't have to spend energy to move things by osmosis. And it's the movement of water across a selectively permeable membrane. So water moves easily across the membrane, but a lot of times other things are not able to move across the membrane. So if we look over here at this picture, we have water on this side and we have salt water on this side. Well, because there's a higher concentration of salt on this side, the water wants to move where there is more salt. So the water is actually going to move from this side to this side. And if we notice, the water is going to dilute out the salt until we have equal concentrations of water. Okay? If allowed, water will move where there is more solute. So water is always going to go where there is more stuff. Water moves towards salt, and then until the water or solute concentrations are equal. Okay, so we give these some terms, and we call them tonicity. And tonicity describes a solution. Describes sort of the osmotic level of that solution. And so there's actually three types of tonicity. The first type is called isotonic. And in a solution that's isotonic, the concentrations have equal concentrations of salt on either side. So if we look here, they have equal concentrations, the same number of salt on this side of the membrane on this side. Which means that water is going to move across the membrane, not the salt, but the water. The water is going to move across the membrane equally in both directions. Water doesn't stay still, it moves across, but it moves equally in both directions so that there is no net movement of water. Okay, so water moves in and out of each side at equal rates. There's no net movement of water in an isotonic solution. Cells in these isotonic solutions, so if I take a cell, here's my beaker, and if I take my cell, and the cell has the same. If I put a cell in an isotonic solution, the cell is happy. Nothing happens to the cell. Water moves in and out of the cell at equal rates, but the cell remains healthy. So if I look down here, here's our isotonic one. If I put cells in an isotonic solution, Here's an animal cell. The water moves in and out at equal rates. The cell maintains its shape. If I look at a isotonic solution of a plant cell, we give it the term flaccid, which means that it'll sort of be a little limp and kind of not very crisp. Water moves in and out at the same rate and nothing really happens to that cell. And that's in a plant cell. So if we look at this, a hypotonic solution, less solute particles than the solution on the other side. So this solution right here is hypotonic. It's describing this one. Well, because of that, water always wants to move where there is more stuff. So the water is going to want to move here. It's going to move away from the hypotonic side. Water rushes away. Because there's more salt over here, water goes where there's more salt, and water rushes away from the hypotonic side. If we put a cell, in a hypotonic solution, the cell will actually burst, and here's why. So here's our hypotonic solution. If I put an animal cell in here, the net movement of water is going to be into the cell. The water is actually going to rush in, and the cell is going to burst. So over here, we'll rupture the cells, we'll destroy them, because so much water rushes in, and it fills out the cells, and it actually explodes the cells. Down here in a plant cell, something different happens. The major water vacuole here fills up with water, and it puts pressure on this outside wall. So we have this pressure that's pushing against the outside wall called turgor pressure, and we give the term turgid. And that actually makes the plant really crispy and makes it stand up tall. So cells in this solution, this is the ideal solution for plant cells. They want to have lots of turgor pressure. They want to have that plasma or that water vacuole pushing up against the outside. This is why celery, if you put celery in fresh water, it'll get a lot crispier because we'll make all the cells turgid, and then that will crispen up your celery. So if you have limp celery, you can just put it in a bucket of cold, fresh water, and that'll crispen it up. Okay, the last one, a hypertonic solution. So this one would be hyper. So here's our hypertonic solution. It has more salt particles than the other side of the membrane. And remember that water is going to rush towards the hypertonic. Right? So water rushes towards the hypertonic. Now if I put cells in a hypertonic solution, the cells will actually shrink. And here's why. If you think about this, I have a beaker. Yikes. Here's my beaker. I'll draw it over here. Here's my beaker, and I put a cell in it. And outside of the cell, that there is lots and lots of salt. And inside the cell, there's far less salt. Well, the water from the inside of the cell is going to come rushing out. So the water from inside of the cell is going to come rushing out. And that cell is going to lose water mass, and it's going to shrivel up. If I look over here, here's our hypertonic solution. We notice we have shriveled cells. So cells in a hypertonic solution shrivel because the net movement of water is out of the cell towards the salty solution. And over here, if we look at this, it's called plasmolyzed. The cell on the inside actually shrinks so far that it essentially dies because that water has been all pulled out of the cell. So here's a great chart for you and all this stuff. So here's the tonicity over here. We can look at isotonic solutions. Here's what happens in iso. It's the same on both sides. Water moves in and out same rate. In a hypotonic solution, it has less particles than the other side. The cell is going to burst because water is going to come rushing into the cell. And in a hypertonic solution that has more, and the cell shrinks because the water rushes out of the cell. So, and then I put up another chart for you here. Feel free to pause and copy this down if you want to. But right here, this kind of lays out everything for you, the types of transport, concentration gradients. So if you want to look at any of those, feel free to go back, pause the video, copy those down. That's important information for you. So if you have any questions about this class, feel free to ask me anytime. That ends our Unit 5 worth of lectures. Have a good day, team.